Hi, my name is Aaron Wang, and today we're here to talk about amylase's effect on yeast fermentation rate in corn stem extract. According to the 2021 Campbell Biology textbook, fermentation occurs when pyruvate, which is made from glucose through glycolysis, is converted into ethanol. In a society where much of our energy is non-renewable, the search for sources of renewable energy is very important. And one of those ways to obtain renewable energy is through biofuels such as ethanol, which is created through alcohol fermentation and are a cleaner fuel than fossil fuels as to produce less carbon emissions. In this experiment, we will use yeast cells to ferment glucose broken down from starches found in corn stem cells, which contain only a little starch according to the 2022 Bio 107 Diet Manual. Although corn kernel cells contain a lot more sugars and would likely produce a lot more ethanol, they are used as food and have many other practical uses, where by comparison, corn stems are usually not used and simply just thrown out. In this experiment, we are trying to see if using amylase as an enzyme to increase the breakdown of starch can produce enough ethanol to justify using corn stems as a more efficient substrate instead of corn kernels, as corn stems are often just thrown out and not used at all. In this experiment, we compared our experimental group against 4 control groups. Each group contained 5 milliliters of substrate and 2 milliliters of yeast. Our experimental group contained yeast, corn stem extract, and amylase. It tested to see if amylase would increase the yield of carbon dioxide production, as more carbon dioxide would indicate a higher rate of fermentation and thus more ethanol being made. We measured carbon dioxide production as it is much easier to measure than ethanol production. Our first group contained yeast, corn stem extract, and a calcium chloride buffer. It tested the amount of carbon dioxide produced if amylase were not present. Our second control group contained water acting as a buffer, stem extract, and amylase. This control group tested that yeast must be present in order for fermentation to occur. Our third control group contained yeast, water acting as a buffer, and amylase. This was the test that sugars and starches are what amylase acted on and that fermentation can't occur without sugars and starches being present. Our fourth control group was our positive control group that tested that fermentation would actually occur if enough starches and sugars were present. It contained yeast, corn starch extract, and a calcium chloride buffer. First, we added the substrates mentioned earlier into 5 test tubes, which were placed in a hot water bath that was 30 degrees Celsius for 10 minutes. After 10 minutes, they were taken out and 2 milliliters of yeast was added. The mixtures were then vortexed and pipetted into a Durham vial that was then inverted and placed back into the test tubes. It was then placed back into the hot water bath where it would remain for the remainder of the experiment. Carbon dioxide production was measured every 5 minutes for 45 minutes. According to this figure, our experiment produced very little carbon dioxide. Control group 4, which was our positive control group, produced the most carbon dioxide at 4.2 milliliters through 45 minutes before it hit its stationary phase. However, the production was not exactly linear. Control group 2 produced no carbon dioxide, while control group 1, 3, as well as the experimental group produced 0.1 milliliters of carbon dioxide over 45 minutes. This graph shows the linear rates of carbon dioxide production of the experimental and control groups. As you can see, control group 3 actually produced about as much carbon dioxide as control group 1 and even more than the experimental group, which was very, very unexpected. Because control group 4 was not exactly linear, we measured the linear rate between 5 and 10 minutes in figure 1, where the production of carbon dioxide was linear, and it had the highest linear rate of carbon dioxide production. From our experiment, we saw that very little fermentation occurred, even with amylase acting as an enzyme. The unexpected result that control group 3 produced some carbon dioxide can result from human error, as air bubbles formed from pipetting the solution into the Durham vial may have been mistaken for carbon dioxide bubbles. The result of not much fermentation occurring could be explained by the fact that stems do not contain a large amount of sugars and starches needed for fermentation. A study done on banana cells in 2014 showed that banana cells contain 3.4% lignin, 4.4% hemicellulose, 35.2% cellulose, and 7.3% ash. Starches and sugars were so low in quantity that they weren't even mentioned. This is a graph of the data mentioned earlier, and the untreated values were what we were looking at. Data from a 2007 study showed that corn stem samples contain just 1.48 grams of starch per kilogram. Compared to things like cellulose, which contained 475 grams per kilogram of cellulose, it was a far lower in quantity than other polymers found in the corn stem cells. This data supports and explains our results, as the concentration of starch was far too low, and as a result, not much fermentation occurred, even with amylase present. In conclusion, amylase didn't have much effect on the rate of yeast fermentation of corn stem extract, as the stem just simply didn't contain enough substrate for it to act on and make a difference. Corn stems are not a more effective substrate alternative for starch breakdown because they don't contain enough starches. In terms of ethanol production from fermentation, corn kernels are more efficient and should be used. An extension of this experiment could be to try and break down the higher concentrations of cellulose inside the corn stems with cellulase enzyme, as cellulose is also a polysaccharide that is made of glucose. Then we could test to see if breaking down cellulose with cellulase from stem extract would result in enough fermentation to make it a more effective substrate for fermentation than corn kernels. Here is my literature cited, and I would like to thank my lab partners Eli and Sonia for helping me with this lab.